This has been an incredible meeting, and I'm glad to be able to participate. Putting my talk together, I was torn because I have two different ways that I would like to be able to contribute or to use a 3D virtual cell. So since I'm trained as a pharmacologist, I always think about how drugs work on a cell and how the cell responds to different disease states. So I'm going to start off by my talk by talking about the how I can contribute to structure-based models. So I use a technique called small angle scattering. This can be done with X-rays or neutrons, and the technique is fairly straightforward. It's a solution technique, so it's very versatile. It can be used for looking at molecules as small as a 1,000 or 2,000 kilodaltons, upwards of megadaltons. So it's possible to make measurements on a variety of different types of molecules. So the technique is shown here where you put your sample in a beam, either an X-ray or a neutron beam, and that beam is scattered by the particles, giving you this radially symmetric pattern here. When you integrate that and subtract out the solvent, you end up with the basic scattering curve, which is the intensity, this is a log scale here, versus the scattering angle, which is expressed in reciprocal units of length. That can be transformed to the length distribution curve, the P of R curve, which is a probability of distances between any two atoms within your particle. So this gives you information about the size and shape. So for instance, this value here where the curve comes down to 0 is the maximum end-to-end length of your molecule. So you get low-resolution, low-resolution information about the size and shape. Now, since your sample contains all your orientations of molecules as well as the various conformations of molecule, you're sampling a very large number potentially of structures. And that's where the modeling becomes interesting and also challenging. The first thing that we can do is if we have a crystal structure or an NMR structure or some other high-resolution structure, we can compare the predicted scattering with our actual experimental data and see how well they fit. That's fairly straightforward. But that most of the problems that we're interested in, most of the signaling complexes, for instance, are much more complicated than that. We don't have single structures. What we have to do is build structures. So one technique we can use is called ab initio modeling, where we use various computer-generated models using dummy atoms to construct shapes that can then yield the scattering curve. The problem is we're taking one-dimensional data and creating three-dimensional models. And obviously, you can create a number of three-dimensional models with any one-dimensional data set that are very different in their shape that can all yield that one-dimensional data set. A more satisfying way, if we have information about the structures of domains, is to put those domains together and create what we call rigid body models. The next step up is if we have a multimeric protein, we can use neutron scattering to tell us information about the organization of that multimeric protein complex. We can tell the positions of where the different subunits are, and I'll give you some experimental details of that in a minute, or an example of that. One of the other challenges that we have is that if we have a flexible model, we have an ensemble of structures, and we now have some software which is requires, I think, further development, but software that we can use to determine what kinds of conformations we might have in our sample. So I've been fortunate to have two outstanding collaborators for the last, 15 years, 20 years, Susan Taylor and Jill Truella. Jill Truella is an expert in small angle scattering, and you've already heard Susan talk. She's an expert in protein kinases and particularly pKa. So we began a collaboration about a dozen years ago to use small angle scattering to look at the differences between the pKa isoforms. And as an example of the kinds of results we have, this is the P of R curve showing the difference between the type 2b and type 2 alpha isoforms. And you can see that they have very different sizes and shapes, even though they have very similar molecular weights, very similar domain organizations. So this has been an ongoing project that's been very interesting. This is one of our first models where we took crystal structures that you've already seen in Susan's talk and compared the type 2b and 2a. This is a fairly simplistic model. We're trying to get more sophisticated. This is another example of the kinds of things that we can do with small angle scattering. This is a student that was in Susan's lab, Cecilia Cheng, and she wanted to investigate the effect of mutations in the R1 alpha subunit, as well as what happens when you truncate one of the cyclonucleotide binding domains. And so she did a series of experiments using small angle scattering, modeled those, and made some conclusions regarding the dynamic properties and the effects of those mutations on the dynamic properties of the A and the B domains. And then this is an example of how we use neutron small angle scattering, and this is with the type 1 holoenzyme. These are the data, the scattering data. The way you do these experiments is you deuterate one of the components of your complex, and then you put the sample into different concentrations of D2O-H2O, so you do a solvent contrast series, and from that data you can resolve the individual scattering contributions of each of the subunits. And so in the P of R curves here we see in this case the C subunits. There's one peak here which represents the scattering within one of the subunits, actually both of them combined, and then the peak out here is the scattering, the interparticle distances resulting from the scattering between these two subunits. So we get information about how far apart these subunits are. Where there's also cross-correlation function that we can determine where these are in the complex. We can also compare the scattering that we get of the, in this case, of the free R subunit without the catalytic subunits, and from this we concluded that the R subunits became more extended when they bound the C subunit. And these are some of the models that were generated from those data. So we still have lots of challenges. A lot of the data, the challenges have to do with shape restoration. And in the interest of time, I'm going to move on to the second part of this. And this comes from my usage of structural models in trying to educate pharmacists, medical students, graduate students. So I'm an editor of a textbook, and this is a figure in the textbook that I helped to prepare. It's kind of a standard drawing. You probably have all seen this. This is how G protein couple receptors work. And so I've been trying to build animations that can provide information about how these work. So do I start the animation from up there? Oh, there we go. So this is, I give a lot of credit to David Goodsell for providing the insights in how to be creative in providing just the right amount of information that can provide a medical student or a pharmacy student or a graduate student information about how these structures work in a much more interesting and informative manner than you might get in a 2D. And I'm going to skip over a couple of these slides here, but I can show conformational changes, we can show some of the size and shape interactions, and those sorts of things are very useful. So going back to the drug action component, what do we need to be able to understand how drugs work in a 3D cell? Well, we need to have lots of data. The PDB provides a lot of that, but there are other databases. We need to have information about the drugs, pharmacodynamic data about binding constants, selectivity, that sort of thing, information regarding the organization of structures within the cell, structural dynamic data, computational resources, and personnel resources. So I think I'm going to end at this point, so I got the stop sign. Thank you very much.